Okay, I want to do kind of a quick review of the last two slides from the last PowerPoint or last video. So we have the word phenotype. Phenotype means physically the way you look. So pheno, physically, if that helps you to remember that. So if I asked for a phenotype of your hair color, you'd give me the color of your hair. Genotype would be your genes. Technically, we can't see genes. Like I mentioned earlier, we're using letters to represent them, and there's only really three options. Big letter, big letter, little letter, or little letter, little letter. And it does not matter what letter you use. You could use A to represent 100 different traits. It does not matter what letter you use. On a few slides back, I just picked random letters for those pea plants. So I picked R for seed shape. I picked T for seed height, plant height. I did not have to use R, I did not have to use T, and for every single trait on that slide, I could have used the same letter. Using the same letter, though, can get kind of confusing. You can use whatever letter you want to use. I also recommend that you use letters where the capital letter and small letter look very different. So I do not want to see any of you using W's, or P's, or C's, sometimes Z's. Look the same, big versus little. Even if maybe my worksheet says use letter P, don't do it. Use letters that your capital and your small look different. Because if I can't tell the difference, then you're going to get it wrong. Then we have heterozygous and homozygous. And I've used the word heterozygous in the last video. Heterozygous is where two alleles are different. And remember, allele is the form of the trait, and you get one allele from mom and you get one from dad, which is why we always have two letters for every trait. The heterozygous allele always shows up in the F1 generation. The F1 generation is always heterozygous, meaning capital letter, small letter. They're different. Homozygous, if the two alleles for the trait are the same. So you got the same allele from mom that you did from dad. Whether it is dominant or recessive, the dominant would be capital letter, the recessive would be little letter, little letter. The P1 generation is always homozygous. It's always homozygous dominant crossed with homozygous recessive. Dihybrid versus monohybrid. Mono means one, hybrid, a child with different genes than parents, and a cross, parents having sex. So crossing parents who differ in only one trait. And when Mendel first ran his experiments, he just looked at one single trait. He crossed plants that were tall with short. He didn't care about the other traits in that plant. There are some, but he didn't look at those. He crossed pure round versus pure wrinkled, and that's all he looked at in that plant. So he crossed, let's say, big R crossed with little r. After he did all those experiments, he then wanted to know does the gene for height control the gene for seed shape? Does being tall determine if it is round or wrinkled? So he did something known as a dihybrid cross, where you're crossing and looking at two traits at one time, so you're going to have two different letters. For the example here, you'd have maybe the A representing height and the E representing seed shape. And you would still start with P1. You'd have pure dominant crossed with pure recessive. So big A, big E crossed with little a, little e, little e. Two different plants, but we're looking at two traits, the A trait and the E trait, or height versus seed shape. He wanted to know, would they affect each other or not? We are going to start out with just monohybrids. We're going to do dihybrid crosses at a later time. I want to do just another quick review here. So we had Mendel's P1 generation. So we'll do seed shape again. There's that cross. You have to show the X. So pure dominant crossed with pure recessive. This would be known as, I'm just going to abbreviate the homozygous dominant crossed with the homozygous recessive. Homozygous dominant crossed with homozygous recessive. I'm using some of those vocabulary words. When those two parents had babies, the only thing that they could have is a heterozygous plant. But its phenotype for this heterozygous plant would be round. So again, we cross pure round with pure wrinkled, or homozygous dominant with homozygous recessive. We got the heterozygous plant that looked round. The next thing Mendel decided to do was to cross his F, or the babies he got from P1 with each other. He called them those F1 generation. So he took the heterozygous round, crossed with a heterozygous round. And the babies that were produced from this generation were known as the F2 babies. And when you cross heterozygous round with heterozygous round, you get the following. You get a homozygous dominant round, you get a heterozygous round, and you get a homozygous recessive wrinkled. Those are the plants that that will be produced, and 25% of the time there will be homozygous round seeds, 50% of the time there will be homozygous wrinkled seeds, and 25% of the time there will be homozygous wrinkled seeds. I think I said that wrong. So here, this happens 25% of the time. You have the heterozygous round 50% of the time. And you have the homozygous recessive or the homozygous wrinkled 25% of the time. And we're going to find out how you know those percentages here in just a second. After I clear, okay. So you have the parental P1 generation, the parental generation, and a breeding experiment. The F1 is the first generation of offspring in a breeding experiment. The F stands for the filial. Then you have that F2 generation, which is the second set of offspring that were produced. So P1, F1, then F2. And then here again is just a list of his traits and actual numbers he got when he did his counting of his seeds. Probability is the likelihood that a particular event will occur. That's where the whole 25, 50, 25% comes in. You have to know a little bit about probability. So circle the letter of the probability that a single coin flip will come up heads. Well, you should know that if you have a coin, you have the heads versus tail. You have two options, so it's going to be 50%. 50% is your answer, or C. 50% of the time should be heads, 50% of the time should be tails. That's not always what happens, but that's the chance you have every time. And then how do geneticists use Punnett squares? They're going to use the Punnett square to determine all possible options for the baby. All possibilities for the baby. Punnett squares, what are those? You're going to find out in a minute. But we use Punnett squares in genetics to determine probability, to determine the possibilities for the children. So here is an example of Punnett square. this would have been from a cross that looked like this. So we would have had, and again, this is not my slide, so if they're using peas, I would not recommend using the peas. So there's the cross. To me, it looks like you have a homozygous dominant crossed with a homozygous dominant. And it is showing you across the top would be the female gametes or eggs. Across the bottom you have or across the side you have the male gametes or sperm. So let's say this was the female right here. This is your male. And you remember with your eggs and the sperm, you only get one of every gene. You get half. So if this was a gene in somebody, in the female, the egg could either get this option or this. Same thing with the male producing sperm. If this is the gene, when this male made sperm, one sperm could get this gene, one sperm could get this gene. So it's all possibilities. So for the female, we put one of the options here, one of the options here. For the male, you put one option here, you put the other option here, and then you're just bringing them together, like they're showing you here. Well, in this box, these are all going to look the same because we have homozygous going on. Okay. The next slide also shows you an example of this. So you can look at that closer. You can take some time to pause the video to look at that. And then here's more explanation that you can look at. I want to go and do some punnett squares. Okay, we'll start here. You have to make a box. This first person, possibility for, let's say it's a female. If she makes an egg, the egg could have a big T or the egg could have a little t. So I'm going to put a big T here and I'm going to put the little t here. Those are the possibility for her eggs. If this is a male, he has a possibility of having a dominant gene in his sperm or the recessive gene in his sperm. So I'm going to take the big T and put it here. This little t here, and again, across the top and down the sides are the eggs and the sperm. You bring an egg and a sperm together, you get fertilization, and you make babies. So I bring the big T down and the big T over. I'm going to go up here and bring the big T down. And I'm going to bring the little t over. I always write the little t second. Here I bring the little t over. So you can know it's a big T. The little t down. I write the dominant gene in front of the recessive gene. It's going to overpower it. Here I bring the little t down. And I bring the little t over. At this point you should pause the video, practice the other 3 punnett squares, and I'll come around and help you. Here's just another explanation showing red flowers, yellow flowers. A genotype of an allele, it's either red, yellow is the recessive. Again, all genes occur in 2 pairs because you get one from mom, one from dad. And so possible genotypes would be big R, red. Big R, little r, we'd still have a red plant. And then you have the homozygous recessive, which is yellow. Homozygous dominant, big R, heterozygous, big R, little r. Homozygous recessive, little r, little r. Gene expression. Are the following sentences true or false? Homozygous organisms are true breeding for a particular trait. Plants with the same phenotype always have the same genotype. And that's where we'll start our next video. You come up with an answer.